knowledge, and the topic would be prokaryotic cells. So, first question is from 2015. Of the following, the most effective method to kill the bacterial endospores is A. Moist heat sterilization, B. UV radiation, C. Filtration, and D is pasteurization. So, first of all, to answer this question, you have to know what is bacterial endospores. So, whenever a bacteria encounters environmental stress such as pH imbalance, heat shock, cold shock, or dehydration, then in these kind of environment, it tries to adapt itself in order to survive the harsh conditions. What it does, it so this is a vegetative cell which will be present in case of a normal environment. So whenever there is a stress condition, it tries to form an endospore. And how does it happen? It happens so the bacteria divides asymmetrically, and in this asymmetric division, all the DNA, but like most of the DNA of the bacteria, the essential proteins, and all are concentrated in one pore, which forms the fourth spore. And through various uh, processes, finally endospore is formed, and around that there is a coat to protect the endospore, and then the spore matures, cell lysis happens, and the endospore is released. This is the structure of the endospore, where you can find the DNA, the spore wall, inner membrane, cortex, as well as the exosporium, that is the endospore wall coating. So this is the basics about endospore. Now they are asking the most effective method to kill the bacterial endospores. So now for killing the bacteria or the any kind of microorganism, we employ sterilization or disinfection. So no, now these two things are very widely used and commonly they are misjudged. Sterilization means it is a process by which any article, surface, or medium is made free of all the microorganisms, either in the vegetative or in the spore form. Okay, all the microorganisms are killed. But in disinfection, it is capable of killing only the disease causing or any infectious microorganism, but it cannot kill the spores. And all organisms may be killed, but may not be killed, but the number is reduced to a level that is no longer harmful. Here, all organisms are killed, but here the number is mostly reduced, which is so that it is no longer harmful to the health. Now, when we come to physical methods of sterilization, which are the most commonly used methods of sterilization, we have heat sterilization, and in that also we have moist as well as dry heat. So, in moist heat sterilization, we can do it at a temperature below 100, at 100, or above 100 degrees centigrade, and in dry heat also we have red heat or incineration and infrared radiation with hot air oven. Filtration is another process of sterilization, which includes passing the medium like the solid, liquid, or gas through a filter, such that mostly we use the point 0.2 filter that does not allow any bacteria to pass through it. And so we can sterilize liquids and gases. Then we have irradiation, which can be with UV or ionizing radiation, and some other less commonly used are sound, pressure, and sunlight. Now, sterilization by heat is the most commonly used, and we have moist as well as dry heat. So, moist heat is the most effective and efficient. It kills the organisms by denaturing the enzymes and proteins, and spores are also killed by exposure to moist heat at 121 degrees centigrade for 10 to 30 minutes. In dry heat, also, most of the organisms are killed. The action by oxidative destruction of the cell constituents, and spores are killed at 160 degree for 1 hour. So both of them are capable of killing the spores. But here you can see that only within 10 to 30 minutes all the spores are killed, whereas in dry heat we have to keep it for 1 hour. So in terms of effectiveness, moist heat will be the most efficient in killing the spores at a less time and at lower temperature. So when we come back to the question, we saw here, if we see the options are moist heat sterilization, filtration, UV irradiation, and pasteurization. So we talked about moist heat sterilization and filtration. In filtration, I also mentioned that we have to the lowest we can filter is size is of 0.2 micron. So this is the lowest filter side, but the endospore is lower than that. The size of the endospore is even lower than that. It comes around zero point 0.1. So it can pass through the filter, and therefore it cannot, the filtration cannot kill the endospore by any means. Now, next we have pasteurization. So, pasteurization is heating, mostly done for milk. So, it is heating at a temperature of 60 to 70 degrees centigrade. So, that also is not sufficient to kill the endospore. And lastly, we have UV irradiation. So, what happens in UV irradiation is that whenever we pass UV light through the bacteria and virus, it creates DNA breaks in the DNA. That is how it kills the bacteria. But endospore, if we see that it is a dormant, tough, and non-productive structure, the primary function is to ensure survival of the bacteria through periods of environmental stress. Therefore, they are resistant to ultraviolet and gamma radiation, desiccation, temperature starvation, chemical disinfectants. So here clearly it is mentioned that they are resistant to ultraviolet and gamma radiation. So therefore this method is also ineffective. So the only method which can kill the bacterial endospore is moist heat sterilization. So option number A is the right option.